Welcome students, I am Dr. James D'Souza, Assistant Professor, St. Xavier's College. Today's program is for Bachelor of Science, third year, subject Botany, semester 5, course code BOC 107, course title Microbiology and Plant Pathology. Title of the unit Plant Disease Management. Module name Development of Transgenic for Disease Management. Coming to outline, in this module we'll be discussing about plant disease. Development of transgenic for disease management. At the end of this module, student will be able to explain the plant disease. They will also able to describe the different developmental of transgenic for disease management. Coming to introduction. Now, before the introduction, first we should know what is a plant disease. Now, in plant disease, there are three components: pathogen, environment, and host. When these three components interact, disease is formed. Now, how we identify the disease plant? We can clearly see the signs and symptoms. There are morphological changes, and also there are physiological processes which are affected, which help us to distinguish the disease plant. Now, to control the disease, there are many methods. There are biological methods, chemical methods, there is IPM system, and there is transgenics. So, today's module will be discussing about transgenic for disease management. Now, first we should know what is transgene. Now, transgene is that genes which is artificially inserted to improve the plant trait, and the process of insertion is called as transgenesis. Now, during this process, introduction of exogenous DNA into genome is done to maintain a stable and heritable character. Here, modification of DNA using genetic engineering techniques is done to introduce new trait or the character which will benefit the host plant. Now, what are these transgenic plants? The plants whose genome is altered by adding one or more transgene. Now, coming to the history of transgenic plants, these are some of the example of the transgenic plants which was developed, mostly the food crops. Now, this was developed to increase the resistance of the plant to the pathogen and to unsuitable environmental condition, reduction of spoilage, or improving the nutrient profile of the particular crop. Now, we will see the example. In the year 1982, antibiotic-resistant tobacco plant was produced. In 1987, insect-resistant tobacco plant was produced. Next, to increase the shelf life, there was a tomato variety which was produced in 1994, which is called as Flavor-Sour. And again, herbicide-resistant tobacco plant was produced, which was resistant to bromoxynil herbicide. Now, transgenic maize and soybean resistant variety was also developed, which was again resistant to herbicide 2,4-D. They were developed by inserting a bacterial aryl oxyalconate dioxygenase gene. Next, Bt toxin, which was isolated and used for TI plasmid mediated transformation of tobacco, cotton, and tomato plants. Now, why this transgenic is required? Now, by using this technique, desirable genes can be inserted. Now, these genes provide features such as high yield, pathogen resistance, and improved quality. By using this transgenic, they can generate more useful and productive crop variety containing new combinations of genes. Now we will see in the pictorial form why this transgenic is required. The first point is pathogen resistance. We can help host plant to develop resistance, which will help them to protect from pathogen. Then, to avoid biotic stress, it helps plant to increase their tolerance to biotic stress. Then, we develop transgenic to improve the nutrition quality. When we say nutrition quality, there are certain plants like certain varieties of soybean was produced was transformed to increase the oil profile. Similarly, golden rice was produced. In that rice, there was high content of vitamin A. Coming to industrial product can be also increased. In case of tomato, that is enhanced shelf life, here tomato was used to increase their shelf life. Now transgenic is also used to provide herbicide resistance. Now which are the methods? Now, there are many methods. First, biological methods. Under that we have agrobacterium mediated gene transfer, plant virus vector. Then we have physical methods, electroporation, microprojectile, microinjection, liposome fusion. Under chemical methods, we have polyethylene glycol mediated and diethyl aminoethyl dextran mediated. Now in this module, we will be discussing about biological method and physical method. Coming to electroporation, here short pulse of high voltage are applied to protoplasm, which make temporary pores in the plasma membrane, which increases permeability and facilitate the uptake of foreign gene. Next, we have microinjection. Here, DNA of interest is taken in microinjector and then delivered inside the cell. Now, this method is successful in tobacco and alfalfa plant species. Maximum 40 to 50 protoplasts can be microinjected. Next, we have biolistic method. It is also called as gene gun or microprojectile bombardment method. Here, DNA is bound to tiny particle of gold or tungsten, which are shoot inside the plant tissue. The particle penetrate both the cell wall and the membrane. DNA separates from the metal and is integrated into plant genome inside the nucleus. And this method is successful in many monocots, example wheat and maize. Next, we have agrobacterium mediated gene transfer. Now, this is a soil-borne bacterium which causes a crown gall disease in the plant. So, this is used. It is used as an efficient vector of DNA because it has ability to transmit a fragment of its large plasmid into a nuclear genome of infected cell. Now, this fragment is called as tDNA, which contain transfer genes. The transgenic agrobacterium is then sent to the transfer the delta endotoxin protein into the plant cell chromosome. Next, the plant cell is cultured and grown into the whole plant whose cells contain this toxic protein. The transgenic resistant plants then produce seeds expressing the insect resistant trait, which can be commercialized. Now, this procedure is also used to transfer Bt delta endotoxin gene to many plants, including cotton, tobacco, tomato, and potato. And now, this is the example of transgenic cotton, which was developed by using Bt toxin. Now, this some years back, in some parts of country, there was a severe problem where these cotton crops were damaged. To control this, what they did was they introduced this method, that is transgenic method, wherein they took a gene from bacterium and they inserted this gene in this resistant variety of cotton. Now, this gene was able to produce insecticidal protein. Now, this insecticidal protein was in crystal form. Now, the insect which was attacking this plant was bollworm. Now, this insect was attacking the young part, the floral buds of this plant, which was causing premature opening. Now, to protect this plant from this insect, this gene was inserted, and this gene was producing these insecticidal proteins in crystal form. Whenever this insect was feeding on that protein, this protein was activated due to the acids present in the guts of this insect, which helps to kill this insect, thereby protecting this cotton plant. Now, what are the advantages of transgenic plants? Resistance to the pathogen and diseases, it enhances the nutrient composition and food quality, it increases the crop yield, it reduces the cost of food or drug production, reduce need for pesticide, greater food security, and medical benefit to the world growing population. Coming to limitations of transgenic plants, damages to the human health. And certain individuals they face allergies and a change of nutrient values. Then damage to the natural environment, that is crop to weed gene flow. Now here there is one disadvantage wherein suppose if you don't track a transgenic crop, there are chances that due to natural flow the gene might go into the weed, which might become nuisance later on. Next, disruption of current practices of farming and food production. Next, disruption of traditional practices in less developed countries. These are my references. Thank you very much.